Well, since urine is basically water with other things dissolved, especially sodium, this whole tutorial is kind of dedicated to the water and sodium reabsorption. Sodium, as we mentioned, can be reabsorbed throughout the whole length of the tubule with the exception of the descending limb of the nephron loop. Water is going to follow by osmosis, which is a very passive movement. And the passage of water or the osmosis of water is dependent upon the sodium reabsorption. Water, though, has to pass through these little channels. The channels are known as aquaporins. These aquaporins are pretty much found throughout the whole tubule. There's lots of them on the proximal tubule. The only problem is that the collecting duct usually does not have aquaporins. Unless we need to concentrate urine, that means that the body will increase the levels of antidiuretic hormone in the plasma, and in the presence of ADH, these aquaporins are going to be expressed, leading to the increase in the osmosis or the reabsorption of water. So we'll see how these processes work. So we'll first talk about the sodium reabsorption in the proximal tubule. So let's take a look at the anatomy of what we're looking at. So this right here is the lumen of the tubule. This is where the filtrate is. These are the epithelial cells of the tubules. That's the apical membrane. And these are the tight junctions between the cells. And this is the basolateral membrane that faces the interstitial fluid, and the peritubular capillary would be right here. So in order for sodium to be reabsorbed, it would have to pass through the apical membrane and the basolateral membrane into the interstitial fluid, and then from there it will be absorbed into the peritubular capillary. So throughout the, you'll find throughout all of these processes that sodium will pass the basolateral membrane with the help of the sodium-potassium ATPase, so that sodium-potassium pump, but it differs on how it crosses the apical membrane. So in the proximal tubule, sodium is actively transported into the cell through active transport, and that, because sodium, as we mentioned, is kind of like a friendly ion, it could usually sometimes co-transports other molecules with it. So co-transport would mean to transport the molecule in the same direction of the flow of sodium. For example, glucose. Okay, so glucose can be co-transported with sodium in the proximal tubule, and then glucose would cross the basolateral membrane through its own carrier. And these again are the carriers that can be saturated in diabetic patients, leading to the inability to reabsorb all of that glucose, and glucose eventually will just appear in urine. Sodium can also be active transported and help in the secretion of other ions, and that process would be known as countertransport. So sodium is going in and hydrogen ions are going out. Okay, so for the sodium reabsorption in the proximal tubule, again, two different things happening at the apical membrane. Sodium is going in through active transport, either to co-transport other substances like glucose or to counter-transport other substances like hydrogen. At the basolateral membrane, though, it is always going to be the sodium-potassium pump. So what is the mechanism of sodium reabsorption in the ascending limb? Remember, the descending limb does not allow for sodium reabsorption. It is not permeable to sodium or any other ions. So we will move on to the ascending limb, and again, it would have to cross both membranes, the apical followed by the basolateral. You'll see here that again at the basolateral membrane, it crosses that by the sodium-potassium pump. What about the apical membrane? Well, there's a co-transporter here known as the sodium-potassium 2-chloride co-transporter, meaning that it'll transport sodium, potassium ions, and 2 ions of chloride, so known as the sodium-potassium 2Cl co-transporter. All of these will be reabsorbed from the tubule into the cell, and then again sodium will be kicked out of the cell through the sodium-potassium pump. Chloride will go through its own chloride channel. Moving on to the sodium reabsorption in the cortical collecting duct, meaning the part of the collecting duct that is found in the renal cortex. So in order to pass through the apical membrane, that is, simple diffusion through a sodium channel, and through the basolateral membrane, again, by the help of the sodium-potassium pump. Now we want to understand the coupling of water and sodium reabsorption. So wherever sodium goes, water is going to follow by osmosis. And that's the process that happens here. Sodium is being reabsorbed. That decreases the osmolarity in the tubules. So water will follow sodium by osmosis, either transcellular or paracellular, which is a little bit harder because it'd have to go through these tight junctions. And then that water will just bulkily flow into the peritubular capillaries. Water, though, as it passes by osmosis, it needs to go through these aquaporins. These are channels that are specific for water. And I want to go through the whole process of the tubules and what passes where, when, and so on. So filtration is going to happen over here. These are your proximal convoluted tubules where both sodium and water are reabsorbed at the same rate. So the filtrate at the end of the proximal tubules would be isotonic. As it enters the descending limb, remember the descending limb only allows for the reabsorption of water, no ions. So the descending limb is permeable to water only. So by the time the filtrate reaches the tip of the nephron, it is now hypertonic filtrate. That hypertonic filtrate passes through the ascending limb. Remember, the ascending limb is impermeable to water. It only allows for the reabsorption of sodium and chloride through that cotransporter, the sodium-potassium 2Cl cotransporter. So we are reabsorbing ions without any water reabsorption. So at the end of the ascending limb, the fluid is now hypotonic as it enters into the distal tubule, meaning that if nothing changes, the urine that will end up being excreted will be hypotonic or diluted urine. In order to concentrate that urine, ADH would have to be present. So diluted urine, urine becomes diluted, as we've mentioned, as you see here, as it passes through the ascending limb, due to the reabsorption of all of those ions without the reabsorption of water. In the absence of ADH, that filtrate that has now entered into the distal and then the collecting tubule and won't really change in the absence of ADH, meaning that under these circumstances, we are going to produce diluted or hypoosmotic urine. When we need to concentrate the urine, so let's say somebody is dehydrated or not intaking enough fluids, ADH levels in plasma increase. They will attach to the receptors, leading, they use a secondary messenger known as the CAMP second messenger system, and that is going to increase the number of aquaporins that are found on the apical membranes of the collecting duct. So we'll take a look at what happens on the collecting duct. So there again is the tubular lumen. These are the epithelial cells of the collecting ducts, and that is the receptor for ADH. Remember, ADH is also known as vasopressin. When ADH attaches to a receptor, it activates the adenylate cyclase enzyme that increases the CAMP as a second messenger. You see here that aquaporins are hiding inside of the cell. But when ADH increases the CAMP levels, aquaporins are going to be expressed on the apical membrane. That allows for more water to get into the cell through osmosis and then pass through the basolateral membrane through these other aquaporins. So here you can kind of see the effect of ADH on the volume of urine and the concentration of urine. So in the absence of ADH or during low ADH levels, we are going to produce diluted urine. In the presence of ADH, so in high ADH levels, we are going to end up concentrating urine. You see here that the aquaporins are going to allow for more water to be reabsorbed, and we end up making concentrated urine. Now, what about the hyperosmotic medullary interstitium that I said I will talk about later? Well, this is later. Okay, this is related to those juxtaglomerular nephrons, where the nephron loops are found in the medulla. We call it there's a countercurrent mechanism going on where there's a descending limb followed by an ascending limb. So you can see how the passage of the fluid in these two different currents going down and then going up, hence the name countercurrent. Okay, it helps in the reabsorption of sodium and chlorides in the ascending limb. And because the interstitial fluid in the medulla is hypertonic, that allows for the reabsorption of water in the descending limb. So I want to take a look at the image in order to better understand that. So I'm going to go back a couple of slides. There we go. So this nephron loop will be found in the medulla of the kidney. The interstitial fluid there is hypertonic. So remember when we talked about how urea is passively reabsorbed, although it is a toxic substance that we would have to secrete later. Well, that urea that is reabsorbed, some of it remains in the medullary interstitial fluid, leading to its hypertonicity. That hypertonicity leads to the pulling or the reabsorption of water in the descending limb. So the descending limb that is only permeable to water and allows for the reabsorption of water is due to the hypertonicity of the medullary interstitial fluid because of that whole recycling process of urea. So let's go back to where we were. There we go. Okay. Here you can see the whole process of recycling of urea, where part of it, lots of it is filtered, part becomes reabsorbed. It is a toxic chemical that we want to get rid of, so it will be secreted later down the road, but it's that recycling and that movement of urea where there is a high level of urea in the medullary interstitial fluid that again helps in the increase in the tonicity there, so the hypertonic medullary interstitial fluid. So that's a term you want to be familiar with. It keeps on coming up over and over again. It'll come up in questions as well. So you want to make sure that you understand what it is. Again, it is the increase or the high tonicity in the interstitial fluid of the medulla of the kidneys due to the urea recycling. And it helps in the water reabsorption from the descending limbs of the nephron loops. Okay, so here it is. Again, we see that image. And this is what a kidney looks like. Hopefully, maybe in another class, you are able to dissect an actual kidney. And then I will see you folks in the next tutorial.